Hello, my name's Dave, and I have the pleasure of bringing to you this course of Biology 30 through this online format. I'm hoping that it's as informative and enjoyable as possible so that you get the most out of it, because it really is about what you put in is what you're going to get out. Like I said, my name's Dave, and I've got the pleasure of being your biology instructor as we go through these slides. I've been doing this for 20 plus years, and I've had the pleasure of being with RTD Learning right from the beginning when they opened up. And I've been doing diploma prep every semester, because I love it. You are going to have a great experience as we go through and really emphasize the process. Then we'll come back and do the terminology. But when you truly understand the process, the terminology just comes together, and it's going to be a very well planned and thought out approach to this course. If you have any further questions, you'll want to go to info@RTD Learning, and whether it's myself or Rebecca, who's going to be doing the majority of the online courses, and she's spectacular, make sure to send us an email, and one of our many biology instructors will take a time, answer it for you, and get your answers right back to you. All right, let's get this journey started. The nervous system and the endocrine system make up 25% of this overall course, so 1/4 of your mark. We're going to take a lot of time looking at the anatomy of the neuron, discussing the pathways, and then a reflex arc, finishing with the synapse before we begin the peripheral and the central nervous system and moving towards our senses such as vision and hearing and balance. Secondly, in this unit, we'll cover the endocrine unit. It's one that I find students need to take a bit more time. There's a little bit more memorization with it, but with understanding the process, where hormones are produced, where they're going, whether you have too much or too little of them, will really add some clarification as to what we're doing within each of these parts. Now, as we begin our unit and we take a look at the nervous system, I always start with my students and I just get us to divide our two nervous systems. Our central nervous system located here and our peripheral nervous system located here. And why I'm drawing that line is to show that they're very distinct in what each system does. Some terms that you would have seen within your course. On our central nervous system, I'm going to put the term interneuron. You should remember that the interneuron is an unmyelinated neuron, meaning it's got no myelin sheath, no waxy coating on it. So interneuron, unmyelinated, we're going to call it gray matter. So all these terms should be coming back to you, and we're also going to say does not regenerate. So our central nervous system involves our brain and our spinal cord, but really remember that term. These are the interneurons that are unmyelinated. Whereas our peripheral nervous system, let's add a couple of terms here. First, it's going to have motor neurons, motor and sensory neurons. So already we have a difference. Different types of neurons, these are myelinated. We're going to call this white matter. We're going to say that they regenerate. So all the things that we talked about on the other side of the central nervous system, and we're going to say it regenerates because of a structure called a neurolemma. And I want to put two things for us to remember in addition on this one slide. We're going to put the word slower on our central nervous system. So if you see gray matter or unmyelinated, it's slower. And on this side, we are going to put faster. And we're talking about the nerve impulse transmission. Faster is not good enough. You've taken this course before. This is a diploma prep. It's faster because of a structure called the nodes of Ranvier. Now, I really want you to remember those comparisons. And just a couple other terms to highlight. Somatic means body, and you have conscious control. So if I threw a baseball to you and you caught it, that is your somatic or voluntary nervous system allowing you to move the muscles to catch the baseball. Whereas your autonomic nervous system, you do not think about. It comes automatically. So out of your conscious control. So things like your breathing rate and your heart rate are controlled by the autonomic nervous system. And lastly, you've seen these two terms, sympathetic and parasympathetic. I just want one word to help remember. Sympathetic is for stress. Parasympathetic is for relax. A key thing about this slide is understanding the neuron is a specialized cells that help us to receive information both from our internal and our external environment. So that's critical. It's monitoring both what's happening within our body and all the stimuluses that are happening within our external environment. As it says, the whole job of this is to maintain homeostasis. Homeostasis is the body's equilibrium. Now, how does it do it? I really like this term electrochemical. So we're going to look at this, electro and chemical. It's got two components. When we are talking about the electro, we're going to be focusing on the sodium and potassium movements. And we're talking about the chemical, we'll be looking at neurotransmitters. So very critical component of our nervous transmission is the electrochemical component within each of our neurons. We'll also study the action potential graphs in a few upcoming slides, and it'll identify for us that just like electricity, our neurons can only fire and move the current in one direction. So here we have one neuron, and here we have bundles of neurons, and this would be called a nerve. When we look at this slide, we should see without a doubt that we're working with a myelinated axon. And we can see the waxy sheath covering that we talked about previously, and we know that this type of axon is going to be faster, and it's also going to have that ability to regenerate. And the key for the faster, like we noted, is the nodes of Ranvier that we'll do in a little more detail on the upcoming slides. But please always note the electrical current can only go from our dendrite receptors up here to our terminal axons down here. It's one direction in the way that it can move. As we look at this myelinated axon, and this myelinated, it actually would be a motor neuron, we need to understand the functions of each of the parts. So when we look at the actual dendrites, we're up here, and we're talking about the dendrites are going to be a neurotransmitter receiver. So they're going to receive information or stimulus. So look for things like neurotransmitters, acetylcholine. We're all talking the same thing. The dendrite receptors will take this. The cell body is going to be where the nucleus is housed and the normal cell functions take place. The axon is this little wire underneath the myelin sheath, and this is where the electrical current is going to travel down. The myelin sheath, as we've noted before, is responsible for the speeding up the transmission as well as the protection. And like it notes here in our notes, unmyelinated will be our gray matter, myelinated is white matter. The Schwann cells are those specialized cells in the peripheral nervous system, remembering there's motor and sensory neurons in the peripheral nervous system, and these create the myelin sheath. So the big thing is produce the myelin sheath. The nodes of Ranvier are these spaces between the Schwann cells. And these gaps are where the impulse is going to jump from gap to gap, and it's going to be a much quicker process to send the electrical current down the entire axon. And lastly, the terminal branches located here, this is where we have synaptic vesicles that contain the neurotransmitter. So in a few slides, we'll be talking about the synapse, and when we're doing that synapse, we are focusing here on the terminal branches and the neurotransmitters that they contain. I often say with my own students, the best way to get anything understood is to really give it a try yourself. Hit pause right now. Take some time. Labels 1 through 8. Do you truly know the parts of a neuron? As I go through this, we're going to see with the neuron, number 1 is going to be our dendrites. This is where we receive the neurotransmitter. Number 2 is our nucleus. This is where we're going to have transcription and translation that we could talk about in our third unit, where the protein synthesis. 3, our cell body, is where the cell processes normally take place and where all the cell organelles will be found. Not testable on the diploma though. That was much more biology 20. Number 4 is our axon. This is the wire that is going to carry the electrical current. Number 5, our myelin sheath. I'm hoping you got that one. Number 6, because it's pointing to the 2 cells, we'll call those our Schwann cells. Number 7, our nodes of Ranvier, and we understand this is where it goes faster because it jumps from node to node. And number 8, our axon terminals, which contain the neurotransmitter. When we look at this slide, we need to understand that we're working with a pathway that is going to begin up here. That could be a skin cell, and underneath it, D would be our receptors that are taking the stimulus from the external environment. And our pathway is always going to go in this direction. So this would be from an interneuron. These are motor neurons. Notice there's no myelin sheath. That's our gray matter. To a motor neuron down here. What I'd like you to do is just take a moment, pause this. Can you label A through J? Do you know what these parts are? Now, moving on with this slide, the pathway never changes. So please always go receptor to sensory to inter to motor to effector. That is huge. I don't care if it's a baby 2 months within the mother's uterus. If they're hearing something, that pathway is the same. Receptor to sensory to inter to motor to effector. Now, taking a look at this, if we were looking at A, I would put a sensory neuron. B, interneuron. C would be my motor neuron. D is a receptor. E is the nucleus. F are the dendrites. G would be the axon or a Schwann cell. I'd accept either, but it really looks like it's pushing at the axon. H would be the Schwann cell. I would be a node of Ranvier. And J would be the effector. So once again, I'm hoping that you paused the video and did really well on all of those parts. Our body has unconscious or involuntary responses known as reflexes. The key thing about a reflex is I want us to remember it is a pre-programmed response that is found within the spine. So as it says, it's going to bypass the brain initially. So it's something that can happen very quickly. So like it says, in response to touching a cactus needle or a hot pot, something allows your body to react before your brain even has to think about it. On this slide, we see, just like we discussed on the last slide, our reflex arc. Beginning right here, we have our stimulus where it's going to be the receptors that take the cactus needle to transfer that message through the sensory neuron. The key thing being there's a pre-programmed response in the spine. It's going to bypass the brain, which then sends the message via the motor neuron so that the person can remove their hand from that stimulus. So once again, the pathway never changes. Receptor to sensory to inter to motor to effector. Key thing here is we bypass the brain initially. Eventually it'll get there, but it's a pre-programmed response. When you look at this graph, it's critical to understand, and I like to divide it in half, that everything on this left-hand side focuses on the sodium, and notice the language flowing in. Everything on the right-hand side is about the potassium flowing out. You have to note those directions. We're talking about the movement of the ions inside the axon. I always point out you need to understand that at these points here, it's getting more positive. Associate a change in where the graph is becoming more positive. So if it's becoming more positive, we're always going to remember that is because the membrane is permeable and sodium rushes in. So sodium is rushing into the axon. If it's becoming more negative, as we see on the right-hand side of the graph, so it's restoring itself or it's in the refractory period, it's all about, so we'll put more negative. So more negative is all about potassium flowing out. So that's key when you look at this graph, the movement of those two ions. Sodium in, potassium out. Sodium in means positive. Potassium flowing out means it's going to become more negative. In this graph, and the graph that we had before on the previous slide, we are now looking at two stages, resting potential and threshold level. The resting potential is going to be here, where we can look at minus 70 would be approximately where the resting potential for this one is taking place. This is when the neuron is not firing. It's in its ready state to receive a stimulus. So at this point, we see we've got sodiums on the outside and more potassiums on the inside of the axon. It's going to move in a 3:2 ratio, always 3 sodiums in and 2 potassiums out. That 3:2 is critical, especially when we're talking about the sodium-potassium pump. So our resting potential, as we said, is at the minus 70. The threshold, as outlined right here, is the minimum stimulus. The threshold can change. It can change with age. You might become more or less sensitive to things. If you become more or less sensitive, that means there's been a change in your threshold level. Less sensitive would mean that there's a greater distance between your resting and now your new threshold. That could be something at minus 40. So it takes quite a stimulus for your actual neurons to be stimulated. So the resting is all about high sodium on the outside and high potassium on the inside. As we focus on the next stage and we look at our action potential graphs, we're talking about a depolarization. And we must remember everything on this side of the graph is about the sodiums. Notice here, the sodiums are rushing in. That is our key language. They're rushing into the axon. And we're not worried about the movement of the potassium. Like it says, it's closed. So this is all about sodium rushing into the axon. Notice when we look at our graph, it's becoming much more positive. So that is going to generate what we call our action potential, or our maximum depolarization. So our action potential, or right here, our maximum depolarization. But please note, it's all about making it much more positive. So as sodium rushes in, therefore, the inside of the axon becomes more positive. As we noted previously, repolarization, when we revisit that graph, remembering it's all about the potassium. Potassium is flowing out. So our permeability of the axon has changed. It's now permeable to potassium, and the potassium diffuses out of the neuron. So now we have a new balance, more potassium outside than inside the sodium, which is inside the axon. Our neuron is becoming polarized again, and during this whole stage, this blue and that orange at the bottom, you cannot receive another impulse. It's in the recovery stage, so cannot receive another stimulus. So once again, make sure you know your directions. Sodium in, potassium out. Here we have one more slide to reemphasize repolarization. The key thing being the sodium channel's closed, potassium channel's opened, and the potassium is flowing out. And as we know, we're also going to see a hyperpolarization or an overshoot. And this is going to be called the refractory period, which we'll deal with on the next slide. But like I mentioned before, the neuron cannot fire when we're in the repolarization or the refractory. The refractory period, like it mentions in our notes here, is the required time before a neuron can respond to another impulse. So if our neuron is stuck on this side, whether it be in the repolarization or the hyperpolarization side, it's unable to receive another stimulus. The hyperpolarization means we've had more potassium rush in, and we're going to require the sodium-potassium pump to help reestablish it back to the resting membrane potential. So if you went for freezing at the dentist, I know that needle hurts when it first goes in, but it basically traps your neurons in that hyperpolarization state so that they can't fire again, and thus the freezing works while the dental work is being done. As noted on the previous slide, the refractory period is the recovery time required before it can respond to another impulse. And we emphasized how it's that negative down here in the hyperpolarization. And the key thing we said was the reestablishment to the resting membrane potential with the help of the sodium-potassium pump, always working in that 3:2 ratio. In this activity, we're looking at what is happening with the sodium and potassium in each box. When I teach this concept in my class, I always explain to the students the neuron can only send its message in one direction. So think of it like we're taking a walk, and I ask my students to find this diagram first, where I can see that the positives have gone inside. That's to mimic the sodium has rushed in. So this is where the action is taking place. So if we're going for that walk in this direction, that means we've already passed the refractory period. So that means it's in the recovery stage. We've already had an action potential there. Now we're in the recovery stage. And we're moving towards this resting membrane. That's our next one. So right now it's in the resting stage, awaiting for a depolarization to take place. So you should be able to look at these diagrams and go, right now I understand there's a high negative content inside the axon. The sodiums are represented by these positive signs, and when we see the positives rush in like we do here, that tells me we've gone through a depolarization. So critical to be able to recognize these stages. As noted before, the actual myelinated neuron is much quicker than an unmyelinated, noting that the actual sodium-potassium exchanges will only take place where the nodes of Ranvier are between the different Schwann cells. Whereas if this was unmyelinated, you would have sodium-potassium exchange down the entire length of the axon, so a much slower process. This allows, with our myelinated neurons, for the impulse to bounce from node to node to node, making it much quicker than having it to open up every channel and close every channel up and down the axon. Here we're looking at a concept called the all or none response, and that's exactly what it is. A neuron will either fire or not fire. There's no in between. If we take a look at this first, I would say there was a stimulus here. I would say sodium rushed in, but it was not enough to reach the threshold potential. This is a stronger stimulus, but it still wasn't strong enough to reach the threshold potential. Now notice they're increasing the strength of the stimulus. So this could be like a tap on the hand, and you feel this one because it reached the threshold the neuron fired. Right here, it's an even stronger stimulus, so maybe you're punching your hand, but notice it never increases the actual size of the graph, the all or none response. You can never make a neuron fire faster nor stronger. So if you see on your exam a graph that goes higher, and they're using this concept, they're testing, do you know the all or none response? Like I mentioned, never faster nor stronger. If it fires always at the same level. I'd like you to take a moment and just pause me for right now and see if you can figure out what's going on in this diagram. We've seen images like this one below. Can you figure out what the three stages are going through it? Now, if I was to look at this, I always start with where I know the sodium has rushed in. That's the easiest one for me to find. So I'm going to predict that depolarization is number 2. It's gone in, the sodium has rushed in. The second thing I'm looking for is that arrow. Remember, it's like going for a walk. Where have we been and where are we going? So if we are looking at where have we been, we've already crossed number 3. So number 3 must be recovering, repolarization. And number 1, because we haven't made it there yet, must be our resting potential. So when I check our animations, 2, 1, 3. One of the final concepts when we're talking about the nervous system is taking a look at the synapse. So if I start up here we're seeing that the electrochemical message, so we've had our exchange of the electricity, the sodium and potassiums, has brought it. Here, we are now in the axon terminals. Notice that within the axon terminal, we have these synaptic vesicles that are containing those blue balls. That would be our neurotransmitter. When you think about a neurotransmitter, I need you to ask yourself, is it excitatory? Meaning, is it going to pass its message on to the next neuron? Or is it inhibitory? The next slide that we're going to look at gives us a good graph, and I want us to always associate excitatory with sodium, inhibitory with potassium. Remember those directions. Sodium flows in, potassium flows out. Now, when we take a look at this process, the synaptic vesicles are going to fuse with the membrane, and we're going to see an exocytosis of the neurotransmitter. Right here is my postsynaptic neuron, and these are my dendrite receptors. The moment the blue ball attaches to the receptor, I want you to ask the question, excitatory or inhibitory, so that you can predict the response on the postsynaptic neuron. The one thing that's not on this diagram that we'll just add is you have what we call cholinesterase. And cholinesterase is an enzyme that is going to work kind of like a Pac-Man and is going to work and destroy any remaining neurotransmitter in the synapse. Its job to do that is to make sure that you're not overstimulating the postsynaptic neurons. So after each time it fires, it resets and removes the neurotransmitter so that it can best receive the next stimulus. And like I noted, you should be able to look at this graph. So I'm talking about over here, it's a process called summation that we're seeing, that A plus B. But without a doubt, I want you to be able to look at A and go, I have no doubts that that was an excitatory response. Sodium rushed in. It made it more positive. B, also, here it is, more positive. So it's an excitatory response. So that's all about the sodiums. Remember that. Here in C, I see an inhibitory response. So that inhibitory, it got more negative because potassium flowed out. Then with the A plus the B, you can tell that summation, it finally had enough of a stimulus to reach the threshold potential and the neuron fired. So like it says over here, note whether it's excitatory or inhibitory. So excitatory, inhibitory, and some examples would be Curare, which occupies the receptor site on the postsynaptic neuron, a pesticide. Predict that. If it blocks the effect of cholinesterase, well, if cholinesterase's job is to remove the remaining neurotransmitter, and we block cholinesterase, therefore there will be neurotransmitter in the synapse, and it will overstimulate the postsynaptic neuron. And Botox, like it says, prevents the release of acetylcholine, so it's preventing the release of the neurotransmitter, therefore the neuron cannot fire. It keeps the muscles in the relaxed state. Thus, the Botox causes the skin to become smoother. All right, hoping that you took a good amount of time to go through the questions. Like I've always said, you can't get good at something unless you try it. And let's see if we put all that practice that we did before to great use. Now, in front of us, we have the divisions of the nervous system. And we know that there's two nervous systems. The central nervous system is seen outlined here, and that will make this one our peripheral nervous system. So as you went through, the big things, when you see involuntary, did that word autonomic come to mind? Meaning you've got no control, but it's done for you automatically. So when you put this together, you should have had the peripheral nervous system, number 1, voluntary control, number 4, spinal cord, as we know, is the second part of the central nervous system, is number 2, and autonomic as number 3. So 1, 4, 2, 3. In the next question, the Achilles tendon reflex. There's a few things to look for here. First, the doctor tapped the Achilles tendon of a man and did not observe a contraction. Immediately my brain says, hey, there's a problem with the motor neuron. Something's happening at number 4. The man reported that he felt the hammer strike his tendon. So we've got the strike happening here. He felt it, so the receptors are working, and the sensory neurons are working. So what's most likely damaged? His interneurons and his motor neurons. In CMT disease, we know it's caused by a genetic mutation that results in the abnormal structure and function of the myelin sheath. So what jumps out to me here is that in the myelin sheath, we know that without a doubt, it's going to have a slower and no ability to regenerate. So when we set this up, our best answer for this one would have been C. When you read this question and you see that you hit your thumb with a hammer and you withdraw your hand, you should have the word reflex come to mind. And when I look at these definitions, A, the threshold of the receptor has been so greatly exceeded that it can't process. No way. Remember, if you exceed the threshold, it passes the message. B, neural impulse is so large that the brain is unable to interpret. Remember your all or none. If it's going to fire, it'll fire. You can't make it exceed or be that much stronger. C, perfect. Occurs in the spine where you have that pre-recorded message, and this will allow you to remove your thumb. So 4, our best answer is C. In looking at the graph and the question, it's asking us to find both the resting and the depolarization. The resting, we'll start here, looks like it's about -90. And our depolarization, so this would be our maximum depolarization, I see this as 40. So -90 and 40. Question number 6 is asking us to determine what's happening in the axon from 0.2 to 0.4 milliseconds. So we're looking at the graph right in this range. We know without a doubt, because it's becoming more positive, it's all about the sodium rushing in. So number 6 would be A. In number 7 on the graph from 0.5, so we're looking right here, to 1.0. Notice it is getting more negative. Everything is about the potassium flowing out. This is not a threshold period. That doesn't exist. But we also know that this section of the graph over here is known as repolarization. So 7 being A. So here we're looking at lignocaine, an anesthetic to block pain during dental procedures. It diffuses into the neurons and blocks the sodium channels. So think about your graph. If it's blocking the sodium channels, we are stuck in the resting membrane potential because the sodium can't rush in. So in 8, our best answer is B, into the neuron, thus inhibiting an action potential. In looking at question number 9, there's a few things that we need to pick out. I'm just going to start with the diagram first, looking at these black triangles. I know those are my regular neurotransmitters. They're found within the synaptic vesicles. Number 2 is a cholinesterase. That is that little Pac-Man that's going to come in and break down any remaining neurotransmitter. These white triangles, number 4, I've got no clue. So I'm going to have to read what's going on to see what this is. So it talks about cholinesterase inhibitors such as the drug donepezil. All right. They can slow down Alzheimer's, but they can't stop the disease from progressing. Donepezil has a shape that allows it to attach to the active site on the cholinesterase. All right, so here is what they're showing me, attaching to the active site on the cholinesterase. In the diagram above, what is acetylcholine? That's number 3. And what is donepezil? That's number 4. So 9 being C. How does donepezil affect synaptic transmission? Remember, it's binding to the cholinesterase. Therefore, cholinesterase cannot break down neurotransmitter.